Hello everyone, my name is Katrin Sophie Skelton, and I'll be taking you through this poster titled T-cell infiltration into tumors induced by DPX-Survivac combination immunotherapy as demonstrated by PET-MR imaging in an orthotopic ovarian cancer model. This work was a collaboration between IMV Inc. and the Biomedical Translational Imaging Center in Halifax, Nova Scotia. So, epithelial ovarian cancer is highly aggressive, and one of the major challenges associated with ovarian cancer is that, due to diagnoses commonly occurring at advanced stages, many patients don't respond well to therapies, and disease recurrence is unfortunately common. Now, despite these challenges, novel immunotherapies like DPX-Survivac show promise. DPX-Survivac is a clinically tested peptide-based therapy that drives activated T-cell responses. In this project, DPX-Survivac was combined with low-dose cyclophosphamide, or CPA for short, and the checkpoint inhibitor anti-PD-1. Now, the overall objective of this project was to gain a better understanding of the mechanism behind the DPX-Survivac combination immunotherapy in this preclinical ovarian cancer model. So, moving on to the results, Figure 1A contains an MR image with a PET overlay from both a treated and untreated mouse, and the ovarian tumors are circled in white. Figure 1B below shows the quantified tumor volumes at all three imaging time points, and on average, tumor volumes were smaller in treated mice, with the difference between groups being statistically significant. So, a common symptom of ovarian cancer is the buildup of fluid in the abdomen, otherwise known as ascites. And in this study, ascites presented in both treated and untreated mice. So, Figure 2 contains the flow cytometry analysis of ascites samples collected throughout the study. As seen in Figure 2A, we found that ascites from treated mice had a significantly higher proportion of CD45 positive immune cells, and this could be indicative of an enhanced immune response. As shown in Figures 2B and C, though not statistically significant, untreated mice appeared to have more F480 positive cells and fewer CD4, CD8 positive T cells. Now, really, one of the primary goals of this project was to gain a better understanding of the DPX combination therapy by tracking the migration of immune cells in vivo using molecular MRI. Specifically, we tracked the migration of CD8 positive cytotoxic T lymphocytes, or CTLs, and bone marrow-derived myeloid cells, or MCs. The CTLs and MCs were isolated and cultured from disease and treatment-matched donor mice. Figure 3 simply demonstrates the phenotyping of these cell cultures. Now, Figure 4 demonstrates the quantification of the MC and CTL recruitment to the tumors and inguinal lymph nodes, and recruitment is represented as the cell density, or the number of cells per millimeter cubed of that specific region. And generally, we found that CTL recruitment to tumors was increased in treated mice, whereas MC recruitment to tumors was increased in untreated mice. Additionally, MC recruitment to the DPX draining lymph node appeared to be increased in the treated group. Finally, the last two figures show our flow cytometry analysis of the tumor-infiltrating lymphocytes. Figure 5 demonstrates that tumors collected from treated mice appeared to have fewer CD45 positive immune cells, fewer CD19 positive B cells, and fewer CD4 positive T cells. However, there was no statistical difference in the proportion of CD8 positive T cells. In Figure 6, we found that there were no differences in various dendritic cell subsets or in F480 expressing cells. Interestingly, in Figure 6C, we found that tumors isolated from treated mice appeared to have significantly fewer granulocytic MDSCs. So to conclude, our findings demonstrated that when compared to untreated mice, the DPX-Survivac combination therapy enhanced tumor control, increased the tumor infiltration of CTLs and the infiltration of MCs to the DPX draining lymph node. The therapy increased the proportion of CD45 positive immune cells in ascites and reduced the percentage of CD19 positive B cells and granulocytic MDSCs in the tumors. Thank you for listening, and let us know if you have any questions.